Hello, can you hear me, people? Am I audible? Audible. Good evening. Good evening. I know other. Can you hear me clearly? Yes. Right, people. Paper hai ka? Right. Right, people. We five. I make a 70. Five. Right, people. Today we are not going to discuss paper 5, but we're going to discuss people the relevant theory parts of paper 5. I think from the last class onwards, there was a request to revise meiosis lesson. Meiosis. So we'll quickly then go through meiosis. We'll quickly go through meiosis. We learn people some of the important things regarding meiosis, right? Meiosis and mitosis. Certain important things we discuss regarding mitosis and meiosis. Right. So we talked about the cell cycle. Sequence of events that happen from one cell division to the next cell division, we call that as the cell cycle. Cell cycle. Just give me 1 minute, people. I'll load the past boards. That's then easy for me. Right, people. Cell cycle, cell cycle. Just give me 1 minute. මේ බෝඩ්ස් ටික මට green screen කරගන්න ඕන. නැත්තම් මේකට දුන්නක් කමන්නේ. මේ pictures bundle එක Telegram එකට දෙන්න. ඔව් දෙපැත්තටම කොකට හරි කමන්නේ. දෙපැත්තට දුන්නා හරි. කොකට හරි දුන්නා හරි මේක green screen කරන්න. Green screen. Right, people. Meiosis, we learn the okay. Oh, hi. Right, people, we learn about this cell cycle. We talked about people, events, phases in the cell cycle. We learn the cell cycle. We learn cell cycle. Come on, remote. Then this. මේකේ මේ ටෙලිග්‍රෑම් මෙන්න මේ ෆෝල්ඩර් එකේ තියෙන පික්චර්ස් ටික මට දෙන්නකෝ එහා පැත්තේ මෙන්න මේ ටික ගන්නවා ටෙලිග්‍රෑම් එකේ මේ පැත්තට not lazy. People, we discuss about cell cycle, different stages in the cell cycle. We learn interface, neither. We talked about interface, and then we talked about photos ටික සම්පූර්ණ බණ්ඩල එකේ මේ විතර දාගන්න. Right, we talked about people different stages, the interface, and then we talked about the cell division, right? Interface and cell division. Are better than mommy board again. My mic, na? Maybe green screen anchor. In the middle now, one. Okay, okay. Right, right. Make a record. The better another. You got the bar? Sure. Right, people, we learn about the cell cycle. So you learn cell cycle. One of you asks a question, right? So we talk about the cell cycle. Cell cycle, we can divide as people interface. Cell cycle we can divide as interface. So two phases in the cell cycle, right? What are those two phases in the cell cycle? We take people this part as the interface. This is interface, right? Interface. And then what is the second one, people? What do you call the other phase? We call the second one as a cell division. The remaining part we call as a cell division. Cell division. Interface and cell division. This part is cell division. Right? Cell division. Now, cell division. Interface you call not M phase. No. Mitotic phase is correct. M phase also correct. Yes. Cell division, or else you can say M phase. Or else what you call people mitotic phase. Not mitosis. M phase. Then interface you learn people, three subphases are there. G1, S and G2. G1, S and G2. Right? That you call as the interface. And then people, the M phase has two parts. M phase is the nuclear division and C, cytokinesis. So what do you call this? Nuclear division. Nuclear division. This part is nuclear division. Right? And then what do you call that? the cytokinesis. This part is a cytokinesis. So when you say cell division, it is actually referring to the nuclear division as well as the cytoplasmic division. Nuclear division as well as the cytoplasmic division, together you call as the cell division. Together you call as cell division, right? I think that part is now clear for you. So we learn, people, the cell which is available here at the beginning of the G1 phase has a nucleus. And then, people, inside we can see cell organelles present. Different cell organelles are there. So G1 is a growth phase. So what we said, people, at the end of the G1 phase, cell is reaching to its maximum size. And then we can see, people, protein synthesis, ATP, energy production, increasing of the number of cell organelles, right? Increasing of number of cell organelles and all, they happen during the G1 phase. Now here, people, we can see that the cells can leave this phase and can go into a stage which we call as a G0 stage. Cell goes into a stage which we call as a G0 stage. G0, right? G0. So we learn, people, most of the body cells are present in the G0 stage, right? So what are the cells present at G0 phase, people? They are non-dividing cells, they are specialized cells. So what happened during this part is the specialization. Specialization occur, cell specialization, and it goes into the G0 stage. The G0 cells are the most of the body cells. They are non-dividing cells, right? Yeah, nerve cells, muscle cells, your skin cells and all. Now, some of the cells which are specialized, right, depending on the need of the body, they can return. So some of your body cells, people, we can see that they are in a process of dividing. You call them as a stem cells, right? So we learn about, people, the plant has meristems. Plant has some dividing tissues are there. We don't have such dividing tissues, right? So meristems, or sometimes we can say cambia. So these meristems are having dividing cells. Then, people, these non-dividing cells, let me show you a dotted region like this. Me take a dotted, those are having, people, G0 cells, right? Those are having the G0 cells, right? Maybe some meristems are present in the tip of the root also. This is also a meristem, root apical meristem, shoot apical meristem, like that. These are the most of the body cells at the G0 stage. G0 stage, right? Same like that in your body. In your body, we can see. In your body, we can see. You don't have meristems in your body, but you have cells in every tissue and organ except in your brain and in your heart. Except in your brain and in your heart, right? You have these dividing cells. They are distributed all over your body except the parts that I have mentioned. You call them as, they are not the meristems, we call them as stem cells. Stem cells. What do you call these, people? What do you call those? We call those as the stem cells. So stem cells present. Stem cells are available, right? Stem cells present. So these stem cells are capable. These stem cells are capable of dividing, right? Stem cells are capable of dividing. So in your body, stem cells present. So we talked about it. So S phase is a phase, people, where the nuclear DNA replication occurs. S phase is a phase where the DNA replication occurs. So if I show you the nucleus on a simple manner with a single DNA molecule like that. So here, when the cell completes the growth Still single DNA molecule present. Then passing through the S phase, people, when the cell reaches to the end of the S phase, the DNA replication doubles the DNA content. So my original cell, right? So stem cells or skin cells? I said stem cells. Mariam. Some of the skin cells are having the dividing ability too, right? Stem. Why do you say that? Skin cells here, people, the DNA content now double. Here, certain amount of DNA present, it's double now there. DNA content double, right? DNA content double here. And then, people, the cell passes through the G2 phase. Cell passes through the G2 phase. And during the G2 phase, we can see that further the ATP production, whatever the replication of some of the cell organelles, especially a special cell organelle replicates, right? Special replication occur there, right? What is that replication, people? Do you know what replicates here? Duplication of centrosomes or the centrioles. That is correct. Duplication of the centrosomes or the centrioles occur here. So that is what happened in this phase, right? And then the cell is now ready for everything, people, for the nuclear division, so that the cell enters into the M phase. We have to discuss what happens during the M phase. We have to discuss what happens in the cell during the M phase. All right. So, M phase. Now, there is another important thing we learn regarding these three phases. We learn something really important regarding these three phases, G1, S and G2. Ne? This is what you learn, important part. Look at this, people. This is a G1 phase cell. This is G1 phase cell passing through the S phase. The cell arrives at the G2 phase. Cell arrives at the G2 phase. Cell comes to G2 phase, right? Cell comes to the G2 phase, right? Now here at G1 phase, we can see, here at G1 phase, we can see, let's take, people, particular, we'll take that example given in the resource book. We'll take, people, how many chromosome pairs? 2 chromosome pairs present. Red long, red short, right? Let's take, people, blue long, blue short. Blue long, blue short. Let's take it like this. Blue long, blue short. Right? So how many pairs? This is a pair. These two are a pair. So 2 pairs present. Right? So we call these two, they do the same job. Red short and the blue short does the same job. So what do you call that, people? Homologous. Homologous. These two are homologous. These two are homologous. What do you call homologous? They do the same job. That is what you call as the 23 pairs of chromosomes you have. Now this is a very simple situation. 2 pairs only. How many pairs you have? 23 pairs. So this is a simple situation. 2 pairs of chromosomes are there. Right? Looks like a smiley face. Really? This one? Right? So 2 pairs are there. Now during the S phase, everything is going to double. So things are going to double. So the red short one makes a copy. Red long one makes a copy. Blue short one makes a copy. Blue long one makes a copy. Blue long one makes a copy. Right? So that is what happens there. That is what happens here. Right? So the available DNA copy. Now, people, we use a special word for the copied DNA, replicated DNA, we use a word. What do you call those replicated DNA? We call them as the cystochromatids. We call them as cystochromatids. So let me take this chromosome out. Let me take the chromosome out. This is that blue color chromosome. And then you learn, people, this chromosome is not alone. Some proteins are attached. What do you call those proteins? Histone protein. Eh? So once it is replicated, once the DNA replication has completed, this is the situation that you see. This is the situation that you see. You can see that the two replicated DNA molecules are attached by protein at the middle, cohesin protein. Cohesin attaches them. Cohesin attaches them. Right? So this both sister chromatids, this whole structure, this has two sister chromatids. Sister chromatids. These are sister chromatids. This is chromosome, neither. Chromosome. Here, two sister chromatids together make the chromosome. Here, one chromosome before DNA replication, one chromosome is referred to the single DNA molecule. After DNA replication, still it is one chromosome. How many sister chromatids are there? Two. Two sister chromatids are there. Ne? Two sister chromatids. Now, these are basics, people. You need to revise these basics because some of you ask here and there some questions from me once I gave you the paper. Ne? So, they are based on this. So, it's good that you ask questions from me, right? Otherwise, you can't ask from your parents. Ne? You have to ask from me. Ne? I taught you these things, right? So, people, this is the situation, ne? Right? This is the situation. Right. So therefore, DNA replication does not double the chromosome number, but DNA replication doubles the DNA content present in the cell. Right. 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 Good. Now, people, we talked about the pairs of chromosomes. Neither. We talked about pairs of chromosomes. Right. Chromosomes available in pairs. You have to understand sister chromatids are not pairs. Sister chromatids are not pairs. Right. Look at this. Look at this. I think I can enlarge this. Can I? Yes. Oh, that's nice. Right. Right. We'll take this, people. This is meiosis. Ne? Where is mitosis? Mitosis. Okay. Sorry about this, but these are some early diagrams, people. Right. So we are talking about the cell cycle. Ne? We talk about cell cycle. Right. Cell cycle. Events that occur from one cell division to the next. One cell division to the next is the cell cycle. Ne? Right. Events that occur from one cell division to the next is a cell cycle. Now, people, cell cycle has some important steps. Cell cycle has some important steps. Ne? What is that? During the cell cycle, cell growth occur, DNA replication occur, DNA separation, what you call the segregation, that is what actually happened in the nuclear division. And finally, people, the cell division, the cytokinesis occur. Those two steps together, what do you call? Those two steps together we call as the cell cycle. Ne? Now we learn this. We learn this now, right? So I think that part is okay. Ne? Let's take an eye. We learn this. We talked about this, right? What happened during the G1 and G2 phase, right? The DNA content is going to double, right? DNA content is going to double, right? Right. DNA content is going to double. Now let's clear this part a little bit, people. Let's try to clear this part, right? What you call as homologous chromosomes, ne? What you call as homologous chromosomes, right? Now, suppose you take this diagram picture, the mom's head, this is mom's head and mom's body. If you assume that the two pairs of chromosomes are formed in the head, two pairs of chromosomes are formed in the body, right? The dad, it is the same, ne? So during gamete formation, you learn half from the mom's chromosome. One of the chromosome her head, one of the chromosome from her body, ne? Come into the egg. Same thing happened with the dad. One of the chromosome from his head, one of the chromosome from his body comes to the sperm. Ne? So once they unite, we get once again pairs. Ne? We obtain pairs. Zygote has therefore chromosome pairs. So it divide in identical manner, and then it had resulted you. Right? So we learned that. Now, people, it's important for us to discuss. It's important for us to discuss the stages of mitosis. Let me find mitosis, people. Don't I have mitosis? Oof. So mitosis is a nuclear division. I think I don't have that. Mitosis is a nuclear division. Mitosis is a nuclear division. Mitosis is a nuclear division. I don't have that. Right? Mitosis is a nuclear division process. Ne? So what happened in mitosis? One single nucleus, it divide and then form people two nuclei. It divide and form two nuclei. So these two nuclei are genetically identical. That means if the starting cell is a 2n1, the resulting nuclei are going to be 2n1s. The starting one is a haploid one. Resulting ones are going to be identical people to the starting one. So therefore, these two guys are going to be haploid 2. Now, people, we learn this mitosis. We can discuss people in some stages. What are those stages in mitosis? Can you tell me? Prophase, prometaphase, metaphase, anaphase, telophase. Ne? Good. So look at these people. We'll take one by one. Let's take one by one. Right? The first one is the prophase. Ne? Prophase events. You tell me now prophase events, people. During the prophase, this is the nuclear envelope. Nucleolus disappears. Ne? Nucleolus disappears. And then the centrioles, replicated ones, begin to migrate towards the opposite directions. Begin to migrate towards the opposite direction by forming the spindle. Ne? They begin to form the spindle and start migrating to opposite directions. Then chromosomes become short and thick. Let me take the simple situation. Ne? Blue short, blue long. Red short, red long. They become visible. Short and visible. Ne? Shortening, thickening, and transforming into chromosomes. So we can see that sister chromatids are now visible. Ne? Sister chromatids are visible right at the beginning, right, of the prophase. So check those events, people. What they say? Chromatin fibers get condensed by shortening and thickening and transform into chromosomes. As a result, chromosomes will be visible through light microscope. And then nucleoli get disappeared. The nucleoli get disappeared. As a result, chromosome get disappeared. Two sister chromatids attached to at the centromere region. And that I have to show. They are attached at the centromere. So if I take one chromosome out and show you that one chromosome, This is one sister chromatid. This is the other sister chromatid. Both are now attached at the middle. What do you call that, people? The point of attachment is a centromere. Now, there is important thing that I taught you. What is that? A chromosome arm. If you take this sort of a chromosome arm, these ends we call as telomeres. These ends are telomeres. They are telomeres. The middle part is a centromere. This middle part region is a centromere. Middle part is centromere. Now, there is an attachment here. A special protein is attaching these two at the centromere. What do you call that special protein, people? Cohesin. That's correct. Cohesin. Cohesin is holding them together. So, these are two sister chromatids are held together by cohesin. So, you learn that. So, you learn it. Nice. Right? So, they are attached here. They are attached there. Right? So, what is the difference between chromosome and chromatid? Now, this one is a chromosome. 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 Chromatin can everything. Chromatin is everything. Net-like structure. Now, you don't see them separately. Now, when come to the prophase, you can see them as separately. Otherwise, they are present entangled. These entangled chromosomes, right? These entangled chromosomes we call as chromatin. You understand that? Is it clear? It's like cotton candy, right? It's like cotton candy. And then you can't see, everything is like net-like thing. But then you take a part of cotton candy and then compress and then make it into a lump, right? And then you make couple of them like that. And then they are chromosomes. So they are, of course, made up of chromatin. Chromatin, right? You understand that? Anisha, is that clear for you? Right? Arineda. So that is, that's the difference. So this is, people, the prophase. So what are the other events we can see, people? Chromosomes, arms of the chromatids attached by a special protein called cohesin. Formation of mitotic spindle begins. Mitotic spindle. Now this is the mitotic spindle. Spindle formation now start. And then people, the spindle form accumulated microtubule complex. Centrosomes move to the opposite poles. And then centrioles are not, right? So those are the events. Now we go to the next phase. What is the next phase, people? Next phase is prometaphase. Prometaphase. Now what happens in prometaphase? In prometaphase, we can see that the nuclear envelope disappears. Nucleolus already disappeared. And then, people, these centrioles are further moving apart now. Centrioles can go across the, what do you call, spindle can grow across the cell like this. Why, people? Because there is no nuclear envelope now. So it can go across the nucleus, right? And then what happens now, people? Now what happens? The chromosomes begin to attach. They begin to attach to the spindle fibers. So there are different types of spindle fibers now, right? There are some spindle fibers, long ones, which extend, people, from one pole to the other. Some spindle fibers are there, the short ones. Some spindle fibers are there, the short ones, like that. Some spindle fibers are becoming kinetoco fibers. Now, something very special happened here. So this is a chromosome, this one enlarged. So you see here, this is the cohesin. And then special two plates come and attach from sidewise. And you call them as, Danisha says, kinetocos. That's good. Danisha, very good. Very good. And then, people, to them, the spindle fibers come and bind. Spindle fibers come and attach to them. So they work like special attachment points of the spindle fibers. Act as special attachment points of the spindle fibers. Which one is acting as attachment points? Yes, cohesin become the attachment points. Actually, people, cohesin, they are winders. they can wind and shorten these spindle fibers. They can wind and shorten the spindle fibers. That is the job of cohesin, right? Cohesin is actually breaking the spindle fibers and then shortening it, right? So this helps actually in the anaphase. You know, people, the chromosomes, the sister chromosomes move to the opposite pole, and someone has to wind and shorten the spindle fiber. Who does that? Kinetoko does that. So this is not the time to talk about it. Anyway, nuclear envelope is disappeared. Nucleolus, they are disappeared. Now you can see spindle fibers are going to attach to kinetokores. And then as a result of this attachment, the chromosomes are now swinging forward and backward. They try to align. Neither. So the chromosomes, some are still not attached. Some are attaching like this. Neither. Some are attaching. So we can see this stage. So check the pro-metaphase events, people. The nuclear lump fragments, chromosome get even more condensed. Special protein called kinetoko attached to sister chromatids in each chromosome at their centromere. Some of the microtubules that attach the kinetoko of the chromosome move chromosome back and forth. Microtubules which are not attached to the kinetoko interact with those from the opposite pole. Right now, the cell goes into the next phase. What is the next phase, people? Next phase is the metaphase. Next phase is metaphase. So look what happened at the metaphase. At the metaphase, we can see, people, right? Metaphase, we can see that these are the spindle fibers. These are spindle fibers. These are spindle fibers. These are spindle fibers. Right? So at the metaphase, what happens? Chromosomes are aligned. They try to align at the middle region of the cell. Chromosomes are going to align. What do you call that? Metaphase plate. Very good. Right? Let's take that, the red short one. This is red short one. This is the red long one, Lamai. This is red long one. This is the blue short one. These are the blue long ones. They all are going to align at the middle region. Still, you can see that the cohesin is at the middle. Cohesin at the middle. Still, you can see sidewise the kinetoko present attaching. Kinetoko present attaching the. Kinetoko present attaching the. Kinetoko present attaching the spindle fibers to it. So this is, people, metaphase. Chromosomes are aligned at the middle. And then the events, what they say, people? Events, what they say? Centrosomes reach to opposite poles. The chromosomes have arrived to a place called as a metaphase plate, which is located in equal distance from each pole. Metaphase plate, equal distant from each pole. Then they say the centromeres of all chromosomes are located in the metaphase plate. At the end of the space, each of the chromosome of the cell get attached to the kinetoko microtubules. You learn that they are attaching during the prometaphase. But now that attachment completes. Right? So attachment is completing here. In the metaphase, the attachment completes. So everything now ready, people, to the separation. Everything ready for the separation, anaphase, right, people? So what happens now? What happens now? They segregate. Some of the spindle fibers which are not kinetoko, non-kinetoko fibers are going to elongate. And now those sisters, poor sisters, are separating from each other. They go away. Poor sisters are separating, and this phase is the shortest phase and the most difficult phase to observe, and this is the anaphase. Anaphase, everything go to the opposite ends. What they say, people, anaphase, chromatids are separated at the centromere, microtubules attached to the kinetoko get shortened and pull sister chromatids towards the opposite poles. Cell elongates as non-kinetoko microtubules lengthen. This is the non-kinetoko microtubule lengthens. And then what happens by the end of the anaphase? Equal and complete set of chromosomes are found at each pole of the cell. Right, then come to the people, last one, the telophase. Telophase, what happens? The nucleoenvelopes are going to reform. Nucleoenvelopes are going to reform. Right, this is what happens here, people. Nucleoenvelope is reforming. Nucleoenvelope reforms. Nucleoenvelope is reforming like this. Two nucleoli are forming. Two nuclei are going to form daughter nuclei. And then you can see, people, red long, red short, red long, blue short, blue long, blue short, blue long are there. The chromosomes now begin to unwind and then transform back into chromatin. So once they form the chromatin, people, I can show you like a net. This is the chromatin. The one who asked the question, you can see it now clear. They don't stay like this. Instead, they go into a network like this. That's chromatin containing chromosomes. Right? Right? So here, this is what happens. Right? Sir, is what last disappeared, first to appear, is what you are asking about the nucleolus. Nucleolus and the nucleoenvelope. Now we say that it's like simultaneously. Check that part, people. Nucleoenvelope reforms around the set of the chromosome. So nucleoenvelope comes first, right? And then the nucleoli reappears. Second is that nucleoli are going to reappear, right? And then spindle microtubules get depolymerized. Spindle microtubules next disappears. And then chromosomes unwind and become less condensed to form chromatin. Two genetically identical daughter nuclei are formed. You were right. Right? That's all. That's all. And then the cytokinesis take place. Cytokinesis happens next by the compressing the cell at the middle. The cell is going to divide into two. Cleavage furrow happens. That we take, people. Now there is a special point then. The cleavage furrow happen after telophase. It says, people, the division of the cytoplasm starts at the end of the telophase. Starts at the end of the telophase. End of the telophase means telophase is over. Right? In animal cells, cleavage furrow forms. In the plant cell, it's forming a cell plate. Right? If you can't remember cell plate, you better check, people, what cell plate is. So then we move to meiosis. Moving to meiosis. Right? Moving to meiosis. Moving to meiosis. We have half an hour for meiosis. Right? Moving to meiosis. Now in meiosis, what happens? Now they have taken meiosis in that example, people. They have taken 3 pairs of chromosomes. So therefore, if you take the meiosis S, and here, now we take the situation with the 3 pairs of chromosomes. 3 pairs of chromosomes. How did you take 3 pairs? Short, medium, long. Right? Short, medium, long. Right? The pairs. These 2 are short pairs. These 2 are medium pairs. These 2 guys are the long pairs. So we can show 3 pairs. How many sets? How many sets, my dear? How many sets are here? How many sets? Very good. Santosh say 2. Shahar says 2. Hana says, Danisha says 2. Very good. Now we learn about the sofa set. Sofa set. This is a sofa set, the black color one with the small seat and the big seat. This is the big seat. This is the sofa set. Previously in mitosis, the sofa set had 2 items. So you bring 2 sofa sets with a big seat and the small seat. couch, and the small ones like that. So here, there are only 2 items. How many sets here? 2 sets. How many pairs available? 2 pairs. But now people, this is like in meiosis, we take a coffee table too. So the sofa set we buy, people, they have a coffee table too. So how many pairs now? 3 pairs are there. Big seats, 1 pair, small seats, another pair, coffee tables, another pair. So what do you call how many pairs now? 3 pairs. How many sets available? 2 sets. This is 1 set. This is another set. Now, similarly, how many sets available in human? How many sets available in human? How many chromosome sets available? How many sets available? 2 sets. How many pairs available? How many pairs available? How many pairs available? 23. Very good. 23. 23, right? 23 pairs. That's good. So you remember that, Ned? That's nice. So people, in meiosis, we take people, everything is going to replicate so that the red short, red medium, red long, all now makes their sister chromatids. So blue short, blue medium, blue long, all are making their copies. All are attached, people. All are attached at the cohesive. All are attached at the cohesive. So this is just the example they have taken, people, 3 pairs. They have taken example with 3 pairs, right? They have taken an example with 3 pairs. That's their preference, no? Right? So we can't do anything with that. So we are talking about meiosis. You have to understand that meiosis has, meiosis has 2 division steps. So one cell divide and then form 2 nuclei and then separate into 2 cells. Once again divide in another and then form another 2 nuclei like that. And then people, the cytoplasmic division, then cytoplasmic division. Then cytoplasmic division like that. So altogether how many nuclei then? 4 nuclei. So ultimately it forms 4 cells. So 2 division steps are there. The first division step we say meiosis 1. Meiosis 1, the second division step we call as the meiosis 2. Meiosis 2. Meiosis 1 has 4 subphases. What are those? Prophase, metaphase, anaphase, telophase. So you call them as prophase 1, metaphase 1, anaphase 1, and telophase 1. And here also we can see 4 subphases are there. 4 subphases are there. Prophase 2, metaphase 2, anaphase 2, telophase 2. Telophase 2. Right? Telophase 2. So how many stages you have to learn? 8 different stages you have to learn. So we are going to begin with people, the prophase. We are going to begin with meiosis 1. We're going to begin with meiosis 1. Right? Pro, meta, ana, telo. Pro, meta, ana, telo. Meiosis 1. Meiosis 1. So I'm speeding people because you have learned these things. I'm just trying to help you to revise the part. Right? No need to go in details now. Right? So prophase 1. So what happened now in prophase 1? Let's take a cell. Prophase 1. Same thing neither. Prophase 1, nuclear envelope and the nucleolus now both disappear because there is no prometaphase. There is no prometaphase. So both happen simultaneously. Right? You can say that the spindle, what happened here? What happens here? Right? Spindle is going to form. Nuclear envelope disappears. Spindle is forming, people. And then a very special event occur. Now we learn chromosome pairs. Now sister chromatid is present but they are not visible. Sister chromatid is present but not visible. Right? Sister chromatid is present but not visible. Remember? Right? So this is the medium size one. Medium. And this is the long one. That's the long one. Sister chromatids are there, people, but they are tightly held with one another. So if you take the other set, right? So these two are pair, these two are a pair, these two are a pair. So now what happened, people? If you look at this, one of this chromosome, if I enlarge one of the chromosome, if I enlarge one of the chromosome, two sister chromatids are held together. Two sister chromatids are held together. They are held together by a lot of cohesin. Because of this tight binding, because of this very tight binding, sister chromatids are not visible separately. Sister chromatids are not visible separately. This is not synaptonemal complex. No, this is not synaptonemal complex. I didn't come for that. What happened next? Chromosomes are going to pair. Now what happened? This guy pair with this one. These two guys are pairing by special protein network, depositing a special protein network. So this guy pairs with this one. These two guys are pairing with each other by depositing a special protein complex. What do you call that protein complex? Synaptonemal complex. That is synaptonemal complex. That happens to the rest of the ones too. These two guys are now pairing to synaptonemal complex. These two guys are pairing also. That is some special event that you didn't observe previously in mitosis. Homologous chromosomes are now pairing. Homologous chromosomes pair by forming special synaptonemal complex. By depositing a special synaptonemal complex, they pair. Right? Now this is having 4 DNA molecules. 1, 2, 3, 4. 4. 4 DNA molecules are there. 4 DNA molecules are available. You call it tetrad. You can call bivalent because it's 2 pairs are there. It's a pair, so it's a bivalent. It's a tetrad because it has 4 DNA molecules. But those are not in our syllabus. You don't have to worry about it. So this is, people, about the first phase. Right? Now what happens? The cohesin dissociate. Cohesin dissociate. When they pair, the exchange is possible. When they pair, what we said, people? The chromosome pieces can get exchanged. Chromosome parts can get exchanged like this. Chromosome parts can get exchanged. What do you call that? That is called as the crossing over. That's called as crossing over. Right? So synapsis, very special event occur with synapsis, that is crossing over. But crossing over become evident. Crossing over is not something visible. But people, at the end of the prophase 1, since the extra cohesin dissociate, what you see, these chromosomes, sister chromatids, become visible as separate sister chromatids. They become visible as separate sister chromatids. At that point, you can see the exchange. At that point, you can see the exchange as an overlap. You can see this exchange as an overlap. Right? They are a bit reluctant to separate. Right? They are a bit reluctant to separate. What do you call this? What do you call that? What do you call that? Charismata. Right? That you call as charisma. Chiasmata. Chiasmata. Right? We call them as chiasmata. Chiasmata present. Neither. Chiasmata present. All right? Chiasmata present. All right? So it's like an overlap. So the end of the prophase 1 cell now enters into next one. What is the next phase, people? Metaphase 1. So let's draw metaphase 1. Let's draw metaphase 1. The special feature, these chromosomes are going to come to the equatorial plane as pairs. Look at this. This is that red long pair. This is that red long pair with the overlap. This is that red long pair with the blue long pair. They comes like this. This is that exchange. This is that exchange with the exchange. And then people, the short, medium size one, the pair of the medium size ones, they also come to the middle. The shortest ones, they also comes like that. Shortest ones comes like that too. Now we learn a special point. We learn a special point here. Spindle fibers, spindle fibers, spindle fibers, spindle fibers, they extend. Spindle fibers extend. Right? Spindle fibers. These are the spindle fibers. Now you learn, people, both cystochromatids of the same are attached to the same side. Both cystochromatids. That means the kinetoco of these two, kinetoco of this kinetoco and this kinetoco attached to the same side. This kinetoco and this kinetoco attached to the same side. That is one of the special point that you can see in, that is one of the special point you can see in meiosis 1, metaphase 1, right? That is very special point that you can see. Next people is the separation. Here, no cystochromatid separation, neither. No cystochromatid separation. So we take the next step. We take the next step, right? We can see with the cystochromatids. We can see with the cystochromatids. We can see with the cystochromatids. Chromosomes move with the cystochromatids. Chromosomes move with cystochromatids. Right? Chromosomes move with cystochromatids to the opposite sides. To the opposite sides. Chromosomes with cystochromatids, they migrate to opposite sides. Right? They move to opposite sides like that. Right. And then comes to the, what is that people? That's anaphase 1, telophase 1. Telophase 1, we can see that the nuclear envelope rearrange. Nuclear envelope, nucleoli, nuclear envelope, nucleoli rearrange. So what do you have? This side red long, red short, blue medium, right? This side I had blue long, blue short, red medium, right? Forget about that exchange. Crossovers are there. I don't know how to show it now. If you like, I can show it, but then I draw it again and again. Right? This is that the crossover part, exchange part. So this is end of meiosis 1. So you get two nuclei. You get two nuclei, haploid. Why do you say haploid? It's N. Because no homologous. Now no homologous. Here, homologous are now in opposite side because they paired and separated. Husband and wife separated. So where is the couple now? Right? Couple is now on the other side. Right? Couples are on the other side. Right? They are separated now. Divorced, neither. They are divorced now. In meiosis, in meiosis 1, haploid cells are going to form at the end. Haploid cells are going to form at the end. Now, people, the cytokinesis will continue and separate the cell into two. Cytokinesis will complete. So, meiosis 2. Yeah. Meiosis 2 is mitosis. Ne? Now, people, this event is over. Now, that event is over. Right? Now, what happens? Next, what happens? Once again, nuclear envelope disappear. Now we are in prophase. Now we are in prophase 2. What happened? This is what happens. Ne? Nuclear envelope, nucleolus disappears. Chromosomes now condense. These centrioles are now moving to the opposite ends. These are now moving to the opposite side. They start to move to the opposite side. And the spindle is now going to form across the cell. They are now moving to the opposite side. They move to opposite side. The spindle is now forming across the cell. Now, when come to metaphase 2, metaphase 2 is perpendicular to the metaphase 1. It's perpendicular. So, what happened there? What happened there? Right? What happened there? Right? What happens there? Right? Chromosomes are going to attach. Ne? Chromosomes are going to attach. This is the red long and this is the red short. This is the blue medium. Ne? Blue medium. That's the blue medium. And here, people, we have the blue long, blue short, and the red medium. Ne? Red medium. These are their cohesin at the middle. They are cohesin at the middle. And then what happened now? Once the spindle attached, they're going to segregate this side. They're going to segregate to the other side. Ne? What do you call that? Anaphase 2. This is now metaphase 2. This is metaphase 2. This is metaphase 2. Right, people? Metaphase 2. Now they are separating to the other side. So let's draw that. Right? These cystochromatids are now moving to the opposite sides. These two also moving to the opposite side now. They are now moving to the opposite sides. The same thing happened here. They now migrate to opposite sides. They now migrate to opposite side like this. They now move to the opposite side like that. This is anaphase 2. Right? This is anaphase 2. So what is condensation that take place in prophase 1? So what is the condensation that take place in prophase 1? Condensation means chromatin forms at the end of the meiosis 1. So chromosomes, again, they have to build up back into the chromosomes. I mean, meiosis 1, end of the telophase 1, chromatin forms once again. So it has to now, to begin the meiosis 2, it has to condense the chromosomes once again. Got it? Hamad Irshad. Hamad Irshad. Clear? Yes. Now the anaphase 2. Anaphase 2. What happens next? What happens next? What happens next is the nuclear envelope is going to reform with the nucleolus. So now, red short, blue medium. Ne? This side also the new nucleus is going to form. The nucleolus, red short, red long, blue medium. And this side also we can see that the same thing happens. Same thing happens, right? So what do you get now? 4 nuclei, ne? Blue long, blue short, blue long, blue short, and red medium. That's it. That's it. 4 haploid nuclei are going to form, followed by people cytokinesis. Followed by cytokinesis. 4 cells are going to form at the end. 4 cells are going to form at the end. That is, my dear, meiosis. That is, my dear, meiosis. Right? I hope, people, this is enough. Right? I hope this is enough. And a big thank for joining with this evening session. The attendance is better than last time. Ne? I'm going to catch the rest of the people also. Right? Thank you very much. Right? And then we'll meet you people. So as a feedback, these BioEves was great to recall the things that I have ignored and have forgotten. Yes, I think that's the whole purpose, right? Right. So how there are 2 centrosomes in one. Yeah. Thing is, at the end of the telophase 1, the centrioles are replicating. We don't talk about that in our syllabus. Resource book has not mentioned that, but anyway, it happens, right? Yeah. Yeah. At the end of the telophase, centriole replication happens, but it is not mentioned in the resource book. That's all. Right? Right? Okay. That's it. So then thank you very much. This is nice, ne? Right, nice, ne? Okay then. See you all. Bye. I'll see you on the Saturday, Saturday morning. I'll meet you there. Right? Until that, have some nice time, ne?